Hello everyone, I'm Mr. Furlong, and today we're talking about chemical reactions with enzymes. In order for life to exist as we know it, there has to be thousands of chemical reactions taking place every second in each one of our cells. In order for that to happen, our cells need some help, and that help is going to come from a small molecule called an enzyme. Now you've seen enzymes in action before. If you've ever put hydrogen peroxide on a cut and it starts bubbling up, that's because of an enzyme that is in your blood. It helped break that hydrogen peroxide down. In our cells, there's all kinds of chemical reactions that are taking place. We can define a chemical reaction as a process that changes one set of chemicals, those are known as the reactants, into another set, those are the products. So in the example of the hydrogen peroxide on your cut, the enzyme catalyze in your blood broke down hydrogen peroxide, which is H2O2, into water, H2O, and oxygen gas, O2. Now all of these chemical reactions that are taking place, we can break them down into just one of two types. The first type are called synthesis reactions. Now to synthesize something means to make. So this is a building up process. So for instance, we eat a lot of sugar in our diet, correct? So here's a glucose molecule, and let's just show the simplified version of that glucose molecule. So there are times where we might need to take several of these monosaccharides and put them together to make a larger polysaccharide. So now we have a larger molecule. We've built up from smaller molecules into a bigger molecule, a synthesis reaction. In order to do that, we needed some enzymes to help put that together. Conversely, we can take a larger molecule and break it down. That's what's known as a decomposition reaction. So we take a look at that word. To decompose means to break down. So again, enzymes can take a larger polysaccharide, like starch, and we can break it down into the individual monosaccharides. So in order for this to happen though, we need two things. One is some energy. Specifically, we call this energy activation energy. Now, there's nothing special about this activation energy. It's just the energy that's needed to start or to activate a chemical reaction. Now, this isn't a lot of energy, but we need something that will kickstart the reaction. We also have to have enzymes. This is what we're talking about, right? Enzymes can do several things. One is that they speed up chemical reactions. So these reactions can happen faster if enzymes are present. It also decreases the activation energy. In other words, we don't need as much energy to have these reactions take place. So think about that. We get more reactions occurring per unit of time, and it doesn't require nearly as much energy. They can be reused over and over again, so it's not just a one-time use. These enzymes can be used multiple times before eventually they start to wear out. Most of our enzymes are made up of proteins. And so we talked about this in another video. It's because of the making of these enzymes that are controlling the reactions that are going on inside of our cells. And they are specific. In other words, they catalyze only one type of a reaction. So because of this, we oftentimes refer to enzyme activity as a lock and key method. So just like one key can open up a particular lock, one enzyme can cause one kind of a chemical reaction to take place. Let's take a look at an example of this. Here is an enzyme. Now I am just using a geometric shape to represent the enzyme. I want to make sure that you get something similar to this in your notebook. We know that enzymes don't really look like this, but it helps us to explain this process. And let's draw some substrates. Now a substrate is a molecule that the enzyme will act on. So I have two different substrates here. And you can tell just by looking at the enzyme which substrate this enzyme will work on. Yeah, that's right, the green one. You know, if we take a look at that blue one, it doesn't exactly have the right shape. Every molecule has its own unique shape. And the enzyme has to have a complementary shape for that. So when you were looking at these substrates, how did you know that this enzyme would break down the green one? Well, when we look at that specific part of the enzyme, what's known as the active site, it has that complementary shape to that substrate, so that the enzyme can simply go over and grab that substrate. This is now what we would refer to as the enzyme substrate complex. Now let's just grab that substrate. But it needs, remember, activation energy. So some energy is going to come along and is going to allow for that chemical reaction to take place. There's the reaction. And then the enzyme can leave. And we now have two products. So imagine if this was a disaccharide. The enzyme can break down that sugar into the individual glucose and fructose molecule. And this would happen in a place like in our intestines while we're digesting our food. And that's all there really is to this lock and key method. The active site on the enzyme will match up with the complementary shape of the substrate. Chemical reaction takes place with the help of a little activation energy, and our new products are formed. And this can happen very quickly. We can measure this time in milliseconds. And then the enzyme is free to catalyze another reaction. So we're going to be practicing doing some experiments with enzymes, and I can't wait to see you in class.